Hi, everyone. My name is Eric Camino. I'm the Vice President of Research here at PPMD. Thank you for joining us this afternoon for this webinar with Solid Biosciences, who are going to give us an update on their gene therapy candidate, SGT-003, which is delivering a microdystrophin with their novel capsid. Just a little bit of housekeeping before we get started today. As a reminder, the webinar is intended to provide information to the public. As we go through the presentation today, if you do have questions, we would ask that you submit them to the chat function at the bottom of your screen. We will have an open Q&A after the Solid team is done with their presentation. We will try to get to as many of those questions as we can in that Q&A portion. Also, we are recording this webinar, so if you have to step away, or if you want to rewatch this later, or you have friends or family who might benefit from seeing this presentation, it should be up on PPMD's website within the next week. Additionally, if you're watching it back then and new questions come up for you, please reach out to us here at PPMD. We'll be happy to either try to answer those questions or pass them along to the Solid team to make sure you can get some answers to your questions. With that, I'm going to stop sharing, and I am going to hand it over to Annie, who is going to introduce the team at Solid that's going to be presenting today. And like I said, after we get through the presentation, we'll have that open Q&A.
That's great. Thanks, Eric, and the entire team at PPMD for hosting us today. I want to express my gratitude to the patient community for taking time out of your busy schedules to join today's webinar. I also want to acknowledge how deeply grateful we are for the brave patients and families who participate in clinical research. As a wise friend said to me not long ago, these boys' greatest commodity is their time. We must use it wisely, we must learn, we must share information to help patients and families make informed decisions and contribute to advancing the field of gene therapy. That is our promise to the Duchenne community. I'm pleased to introduce the members of the Solid Biosciences team that are joining me today. We have Pat Gonzalez, Senior Director of Clinical Science, Matt Harmelink, Medical Director, Therapeutic Area Head in our Neuromuscular team, and Gabe Brooks, our Chief Medical Officer. Next slide. Before we begin, please be aware that some of the information we'll discuss includes forward-looking statements reflecting our expectations and ambitions for the future. Next slide, and I will pass it over to my colleague, Gabe Brooks.
Annie, thank you so much. And let me begin by reiterating the thanks to PPMD, Eric, Kaylin, and Pat Furlong. Thank you so much for having us today, and really for your commitment to creating a space where we can share data that we're very excited about, and thank you, PPMD, to your commitment for really championing innovation for Duchenne and for patients who really need, we believe, better therapies. So Solid Biosciences is a company that is focused on precision genetic medicines for multiple different disease areas, neuromuscular, cardiac, as well as platform technology, and that means really creating components of these therapies that we believe can better able meet the unmet need, and that includes creating novel capsids, new viruses that we believe may be able to better target tissues that are affected. We're focusing today on the neuromuscular area, and in particular on our SGT-003 product, which is targeting patients with Duchenne muscular dystrophy, with the goal of supplying a novel microdystrophin, and hopefully we will see clinical benefit over time as we continue to progress the program. Today, what we'll be sharing is initial early data that looks at our initial experience with safety, as well as what we believe, and we're very excited about, some early biomarker data. Next slide, please. So to share with you more broadly, all of the things that we're working on, we are in the neuromuscular space. Again, talking today here on the program that we're so excited about, our SGT-003 program. We are also working on Friedreich's ataxia, a program that we aim to be in the clinic next year. And then you can see in blue, we have a deep commitment to cardiovascular disease, including a program for an inherited arrhythmia syndrome called CPVT. Just as an aside, you know, the strong commitment to cardiovascular comes back to Duchenne, and we'll speak about this in a minute, because we know that that patients with Duchenne muscular dystrophy have either subclinical or a clinical cardiomyopathy that develops over time. So really our focus on cardiac comes back to Duchenne when we think about how these novel medicines may affect patients with Duchenne. The next slide. So when we are approaching a new molecule, a new therapy for Duchenne muscular dystrophy, what we want to make sure we do is best characterize what's going on with the muscle, because Duchenne is a disease of impaired muscle integrity, leading over time to dysfunction. And so this focus of ours really translates into how can we look at all of the biomarkers for muscle integrity and function, dysfunction, so that as we are testing this new drug, SGT-003, we're better able to describe what potential impacts that drug might have, hopefully for the betterment of the patient. And as we think about muscle, it's more than skeletal muscle. We are thinking about cardiac muscle as well, because we know the cardiomyopathy can lead to heart failure, as well as muscles of respiration. And so we are looking at all of these markers and all of these organs in its totality. So things we're looking at are biologically, what do we see in the blood that might give us some ideas about how the muscle is doing, as well as, you know, comprehensive assessments of muscle function, whether that be through pulmonary function tests, through cardiovascular examinations, through echocardiography, looking at the structure and function of the heart, or as we might expect through functional measures, like things like the North Star or time to rise or a four-step climb. Ultimately, as we better are able to look at how our treatment affects muscle integrity, we believe that this gives us some reassurance and encouragement about the ultimate ability for this drug to be able to affect the clinical health of the patient. And with that, I will turn it over to Dr. Gonzalez.
Thanks, Gabe, and thank you again to PPMD and for the entire community for tuning in today. We are very excited to be sharing these results from, as Gabe mentioned, our first in-human study of SGT-003. This really is the first readout, so we're happy to be providing it to all of you. What I want to go through first is a little bit of background on what is SGT-003. As was mentioned, this is an investigational microdystrophin gene therapy. As it is being developed, it's being developed as a next generation molecule because of the components that we selected for the total assembly of 003. And so we'll go through what each of these pieces are, really what the initial data were that got us to the point of getting into the clinical trial, and then go through a comprehensive assessment that we've done now, passing the day 90 mark in our study for at least 3 of these patients, and that's what we'll be reporting on today. So as an overview for gene therapy in general, and specifically an AAV gene therapy, this is an adeno-associated virus mediated mechanism where we're using a capsid as a delivery vehicle to then be able to actually deliver what it needs to get back to a muscle. And so in Duchenne specifically, we know that dystrophin is what's missing. We don't have a functional dystrophin protein. And through this approach, what we'll be doing is trying to replace that. We use a promoter that is restricted to the locations where we need it, where we need our transgene to be expressed. For Duchenne, that's muscles throughout the body. So really what we're focusing on is something that can have robust, widespread expression, not just in cardiac muscle, not just in skeletal muscle, but all of these muscles, as well as all of our supportive muscles that can also work towards things like pulmonary function, like Gabe was mentioning. What's unique in each one of these different components, however, is going to then drive how therapy is going to act once it actually goes systemically. Because this is a body-wide disease, we know that we need to be able to administer this in a way where we'll be able to impact as much as possible. And so all of these therapies that we're looking at here in 003 specifically is going to be administered through a single intravenous infusion. And so we'll go through a little bit of the details of this and again, get back into the data that then moves us towards that clinical trial. And so with SGT-003 being considered a next generation approach, it really is based on the components, those different pieces that we were talking about in the last slide, of what make up our final product that we are then going to be administering and then seeing expressed in muscle after administration. A key component here is the actual microdystrophin itself. And so I think as we all know, dystrophin is a very, very large protein. And with that comes a lot of different pieces to it, but a difficulty in being able to actually replace it when it's absent. And so we're unable to take the entire full-length dystrophin because it wouldn't fit into something like an adeno-associated virus. So we need to choose which components are going to be put into a version that then can be packaged within a capsid and then delivered systemically. When we were selecting the microdystrophin for SGT-003, we really worked hand in hand and wanted to listen to the researchers, understand what could be the most functional of a transgene when we're selecting these different pieces. And so what we relied on here were researchers such as Jeff Chamberlain and Dongsheng Duan to say, if we're evaluating each of these pieces independently, what can we see that might actually be able to have a different functionality from that full-length protein? And so this key domain here called the nNOS binding domain is what was identified more recently, and when put into a microdystrophin, allowed for the restoration of this protein called nNOS at the muscle membrane. In the next slide, we'll go through a little bit of what it means and what it can do, but at a very high level, its ability to be localized to the membrane because of dystrophin being at the membrane allows the muscle to then get the energy it needs. And there's a lot of different downstream effects that that can have if it is present or isn't present in a muscle. In addition to this binding domain, we need to have the key structural components of a dystrophin protein as well. So on the inside of the cell, we need to be able to bind and we need to be able to stabilize the actin binding domain, so the actin cytoskeleton. On the outside, because dystrophin is really this membrane protein, we need to be able to link up to the membrane so that it localizes where it needs to and then forms that structural link. And that's through what's called the dystroglycan binding domain. And so together, these pieces put together what the total microdystrophin is and that we're using for our approach. With that, as I mentioned, different delivery mechanisms for AAVs are based on the specific capsids that are being used. And so for SGT-003, we actually went back to the drawing board a bit and said, if there are capsids that we know exist right now that do have quite a good propensity to get to muscle, but you need to administer them at such high doses, and you have some off-target distribution as well, is there a way to optimize that? Is there a way for us to be able to get better on-target levels of this capsid to muscle specifically for this approach? And so through a lot of work that was done, especially starting from in vitro non-clinical experiments, then moving into in vivo small animal models and larger, what we saw is that this capsid called SLB101 was able to perform better than AAV9 at first, and this is what it was really based off of, in a way that we're able to target muscle better by using an approach that is rationally looking at receptors that are going to actually be expressed on the muscle and have been shown to be upregulated in dystrophic muscle, which could again become advantageous for the use of this capsid in this specific indication. And so the combination of these two pieces really make up SGT-003. And in addition to what we've seen from our actual ability to target muscle, we've also had some exciting data that have shown us that in doing so, we also can see lower levels getting to the liver. And so as we're administering systemically, we know that we're going to get levels to the liver. We've seen this from other gene therapies as well. But if this is something that we can be decreasing relative to others, that can also be a potential benefit for this therapy. And so we'll be looking at some of these markers too as we go through both our non-clinical data and the clinical data as well. And so I mentioned nNOS, and really what is it doing has been the question here. And so as I mentioned, as a high level, it's required when you have energy demand. And in muscle, nNOS, which stands for neuronal nitric oxide synthase, is able to produce a very important signaling molecule called nitric oxide. And so the role of nitric oxide in a muscle is really, it's a short-acting signaling molecule where when it's properly localized to the muscle membrane, it can then signal the vascular system to result in increased blood flow where it's needed. So if you're exercising and you have energy demand that's needed in a specific muscle, activating neuronal nitric oxide synthase to then produce nitric oxide can result in what you need getting to that location. Unfortunately, when this can't happen, the opposite can occur. So despite your muscle needing more energy and really depleting the energy that it already has, if you can't get more blood flow to the muscle, you're going to go through what's called ischemia, or essentially having a lack of oxygen. When you have this lack of oxygen, that's what can then result in overall damage to your muscle. And so when we then have the damage to the muscle, and that's really what we see in Duchenne because we have the loss of nNOS at the membrane, then that can result in all of these downstream effects, such as muscle turnover, such as fibrosis, and overall muscle damage that will be decremental. So with the approach of including the nNOS binding domain in a microdystrophin, the hope has been that this can add an additional functionality of a dystrophin that's missing when we have the absence of dystrophin in Duchenne. So looking at this overall from a non-clinical basis initially, what we wanted to do is characterize, as I mentioned, first, in vitro experiments looking at differences between this capsid and a first-generation capsid such as AAV9, and then moving into small animal studies and then large animal studies. What we're showing here are a couple of different examples from a large animal study that we did in the monkeys to be able to compare how well is SLB101 getting to muscle compared to 9? And so a head-to-head comparison of these two different vectors using a single equivalent dose between these two, what we can see is that in both skeletal muscle and in the heart, we see multiple fold improvements to each of these tissue types when using the AAV SLB101 compared to AAV9. In addition, on another positive note, when we look at the amount of vector that's getting to the liver, so this is somewhere where we don't need it for Duchenne, we do see that we have about half of that getting to the liver compared to AAV9. So on the positive sides, where we need more, we're getting more. And then where we don't need more, where actually less is better, that's what's also occurring when using the AAV SLB101 capsid. And as one additional piece of data that really got us excited and encouraged to push this program forward, what we did was an experiment where we had packaged in AAV RH74 and AAV SLB101 equivalent constructs of our promoter and our microdystrophin. So these were two different preparations that we made at Solid, and we were able to then administer them, again, single systemic doses to each group of animals, one with the RH74 and one with the AAV SLB101. And what we could see was a very similar trend to what we had seen in both the small animal models as well as in the previous studies of AAV9, where here, looking at the heart and then the quadriceps as a representative skeletal muscle, we see 4 to 6 times higher levels of expression of the microdystrophin in these tissues. So again, supporting what we saw from the biodistribution, here looking at microdystrophin expression itself, one of the final encouraging pieces of data that really motivated us to move this program forward towards the clinic. And so now I'll go through an overview of really what is our INSPIRE Duchenne study, which we are running right now as our phase 1/2 first-in-human study of SGT-003. I'll go through some of the initial data that we have from our biopsies, and then I'll hand it over to my colleague after that as well to go through more of the clinical results that we've seen. And so, as a study overview, INSPIRE Duchenne, as I mentioned, is a phase 1/2 study. This is a first-in-human study, meaning that it's the first time that SGT-003 has gone into patients, and we have gone directly into patients with Duchenne because this is an AAV gene therapy. We initiated the study back in 2024, and initially we had our IND opened in the United States. Since then, we have also been able to expand that further, and the study is intended now to be conducted in the US, Canada, the UK, and Italy. In this study, we are evaluating a single dose level. And I want to emphasize that because when we selected the dose for this study, we really wanted to be aware of both what we're seeing from the non-clinical data to give us a good idea of where we should be dosing, but also acknowledge that with an AAV gene therapy, right now we are not looking at this being a multi-dose approach. It's not something that you can get more than once. And so because of that, the way that we wanted to move into the study was with the dose level that we believed could have an optimal effect. And really that was our motivation to select one dose rather than starting low and going high from there. With this study, we are maintaining it as an open label study because especially as a first-in-human study, we think it's very important to be able to capture as much information as possible, do that within a wide age range, and then be able to also get many outcomes, and as well as objective data, which we'll also be looking at from things like muscle biopsies. Currently, the study is enrolling patients from age 4 to 11, so inclusive of age 11, and both cohorts are currently enrolling. Another emphasis for our specific study is that as we look at things and ways to be able to safely administer an AAV gene therapy, we always consider what are different modes of immunomodulation that you need to be incorporating as well. We wanted to be able to take an approach to make things as simple as possible here for patients, acknowledging that there's always going to be an added burden with everything that you add on to a study. And so we conservatively began the study with only using an increased dose of prednisone as our immunomodulation. So something that's as a steroid, more well known to this community especially. So as we go into the data that we'll be looking at today, initial data that we'll be evaluating are from our day 90 time points. But as we look towards the future and we look and collect more data in the study, we'll be looking at additional key endpoints such as Gabe mentioned, time function tests, the time to rise, additional evaluations of motor function, the NSAA 10-meter walk run, and importantly, objective measurements as well, such as the stride velocity 95th centile, which has been shown to be potentially more sensitive, especially at early time points and in small studies. What we wanted to do in looking at this study design is also learn from others that have come along the way and really pave the way for us. I think every study that comes through, we can learn something from, and we should always be taking into account anything that has come up. And so one of the more recent findings, I think, has been that acknowledging the variability that we have in Duchenne and different levels of sensitivity of some of these evaluations, if we can look at time points that are even longer than 1 year, although that may be more longer than what we want, it could give us that confidence in the ability to actually better pick up a treatment effect. And so this is where we had focused as well, really learning from the community, learning from the results that we've seen so far to be able to optimize our approach. And so going through specifically the eligibility criteria for our study, I mentioned the age ranges, and currently these are dividing the study up into 2 different cohorts based on these ranges, 4 to 6 and then 7 to 11. With that, we are also very cognizant of safety. And so one of the important factors here that we are unfortunately excluding at this time are patients with genetic variants which are specifically deletions in exons 1 to 11 or 42 to 45. And I want to hold on this one for a second and explain it a bit more, because I think this is one where we always want to be very clear on the reason for it and really what does it mean. One thing that's been learned in the last couple of years has been that there is a possibility that patients with certain mutations could have an immune response against a microdystrophin gene therapy. What we've learned as we've come together, especially with other sponsors in the field, has been that if we take a look at the specific mutations that these patients have had, it's typically due to the presence of a mutation within a region that you're delivering of a microdystrophin that you're seeing this immune response. So every sponsor who's running a microdystrophin gene therapy study will likely have different criteria for what these mutations that are included or excluded would be. And these specific criteria are unique to our microdystrophin because these are the pieces that are within ours. Now, we're very hopeful that over time, we're going to be able to learn more with the field and we can evaluate these further. But especially as we start with this study, we want to be very conservative from a safety standpoint and then move forward from there. So in addition to the genetic variants here, we are also looking at things like function. And in the 7 to 11 cohort, one factor that we are evaluating here is time to rise and the 10-meter walk run. The reason for this, again, is that as we move into this cohort where we may be able to see changes over time, if we can further reduce the variability to a point where we know that we can maybe now match up to a natural history comparator, or we can understand really what a trajectory of a patient might be by taking these factors into account, it could help us better interpret some of the early results we see in the study. And so these were really the reasons for including these and specifically within this cohort here. Moving further down, one of the most important things for an AAV-based gene therapy is the antibodies that a patient may have. And so one of the screening assessments that will be done is to evaluate whether there are a presence or not of antibodies. And so we require patients to be negative for antibodies in the study. We also wanted to be able to include patients who may be considering different options right now or evaluating different therapies. And so, especially given the current landscape where we know that there are different therapies that are out there, especially compared to the past, we wanted to be cognizant of this. And so, with this, while we cannot have a patient enrolling who has received a previous gene therapy, we do allow the washout from other therapies such as exon skipping drugs, vamorolone, or givinostat. So a patient would just need to be off of these for a period of 12 weeks to then become eligible for the study so that there's not a variability either from a safety standpoint or from an efficacy standpoint that could contribute to any difficulty in understanding results from this study. And then lastly here, the regimen of patients for steroids to reduce variability, again, wanting to maintain a stable daily oral regimen of prednisone or deflazacort for at least 12 weeks prior to entering. And so from here, we'll move into what we're very excited to report, which are the results from our study as of February 11th, 2025. Both will be summarizing the initial data that we have from the muscle biopsies that have been taken, and then also some of the safety results up to this point in time. I'll emphasize that this is a continually enrolling study, so we're very excited and encouraged by these results. And with that, are maintaining our efforts to be able to enroll this continuously. So overall, in the first patients who were enrolled in the study, the first three are those who have passed the 3-month time point. And these are the three that we'll be evaluating for our muscle biopsy results. These patients range from 5 to 7 years old with a weight of 19.6 up to 27.8 kilos. In addition to these initial three, we have also enrolled 3 additional participants as of this time point and have these follow-up periods that are less than 3 months, which is why we don't have their biopsy results as well. And they are ranging from 6 to 7 years of age as well. As of the time of our disclosure that we made last week, we had also dosed a 7th participant. So that's an additional who we'll add to the list. And as I mentioned, we'll continue to dose in the study. And so we're very excited to really go through the data and highlight the results that we have seen from the muscle biopsies And I'll walk through what each of these different pieces are, both from what the methods are to what the numbers are and how they should be interpreted or can be interpreted. The first is really what we're excited to report, and it's our microdystrophin expression levels by Western blot. And so with a Western blot, what we're looking at is from a total protein that you can get from a muscle, separating it out based on size. So we're looking for microdystrophin completely. And when we're measuring it and quantifying it, what we do is we compare to a normal pool of muscle. And so we can see here that this percentage that we're reporting here is a percent of normal dystrophin. And what does that mean really? And how are these numbers going to be impactful? So what we can see is that across our 3 participants who enrolled in the study, we have a mean of 110% of normal dystrophin. The range is 84% to 135%. So quite evenly distributed around that mean. Each of these participants, when we look at what their baseline values were really negative for any, especially microdystrophin, but any quantification of a dystrophin that may be present here. And so if we looked at these gels that we use for Western blot, we can see that they were blank in the regions that we would be looking at. And as we would quantify them, they would also be approximately 0% of normal. That means that the change that we have from these points were really what we're reporting here at day 90, which are these mean of 110%. And so I mentioned what a normal is. And so when we're looking to understand how do we compare any of these protein levels back to what we would want to see in a muscle, we will take a pool of normal tissues, meaning a non-dystrophic muscle. So a muscle that does not have a variability in the level of dystrophin that it has. So we won't look at a Duchenne muscle because we know that that'll be near 0. We won't incorporate Becker muscle because we know there'll be some variability. And in other neuromuscular diseases, we could see variability as well. So we try to avoid those and get a representative range of what we would expect to see. And this is really how we can interpret this value here, is that 110% of normal compared to the pool that we were evaluating really was right around that kind of normal level, which would be 100% in that pool. Excitingly, as we look at Western blots as one of the key evaluations of the absolute protein levels, we also want to take a comprehensive look at how we're seeing our microdystrophin expressed and what's happening in these muscles. So we use an additional method that's called mass spectrometry to be able to complement this. And so with mass spectrometry, the difference between these two methods is that a Western blot's looking at a full-length protein, so a full microdystrophin. In mass spectrometry, what we're looking at are small pieces of that full protein because we break it up before we can analyze it. What this gives us is the ability to actually look at a unique number. And this is that in this mass spec method, we can target something that only exists in microdystrophin and doesn't exist in dystrophin. And so with that, we can get what really represents a true baseline of 0% that then changes up to this value at the day 90 time point. So we were very encouraged again to see that the results that we have from our mass spectrometry method were quite consistent with the Western blot, 108% of normal by mass spec and 110% of normal by Western. So in addition to absolute protein levels, which we're very excited to see, one of the big things that I'll always emphasize, and as a muscle biologist, I think it really means a lot to me, is that we need to impact an entire muscle. We need to be able to see how widely distributed any treatment effect that we're seeing can actually be able to get across a muscle, because ultimately, any part of the muscle that doesn't get that impact could be susceptible, again, for some of that degeneration and breakdown. So what we were very encouraged to see here was that in evaluating microdystrophin positive muscle fibers, so using here an immunofluorescence method, and this is a representative image here, where the fibers where we have red staining are microdystrophin positive, and those in dark regions are negative. What you can see is that we had a mean of 78% of positive fibers, and this range of 70 to 88% of positive fibers. So really a wide majority of the muscle getting microdystrophin positivity in addition to the high levels of overall protein that we were seeing. So taking this together as a comprehensive evaluation of microdystrophin, really encouraging results from our side for the potential of this to be protective of the muscle and widely distributed through muscles. And so we went through the microdystrophin itself by immunofluorescence, but when we look at muscle, what we can do is take additional evaluations of different factors that either should be at the membrane or can be changing with or without dystrophin. And so microdystrophin, compared to a dystrophin, is going to need to localize to the membrane for it to be functional. And so this is another representative image here of a baseline, one of our baselines without any dystrophin expression. So what we use as our antibodies here to be able to detect whether we have microdystrophin would be common between both a full-length dystrophin and a microdystrophin, which is why we have a value of less than 1%, because we know that Duchenne patients will have some amount of residual dystrophin production. And so these are those revertant fiber levels that you can typically count on a single hand when you're looking at a muscle, a biopsy. So compared to baseline, these changes that we're seeing are truly increases from very low levels, less than 1% to 2%, up to these 77, 88, 70% of positive fibers. Correspondingly, when we look at other markers that are supposed to be at the membrane, like beta-sarcoglycan, what we can see is that as we increase our levels of microdystrophin, we also have a restoration of beta-sarcoglycan at the membrane. And this is another response to the microdystrophin expression because the functionality of a dystrophin being present allows for this dystrophin-associated protein complex to be stabilized. And when it's stabilized, these additional members can also be correctly localized to the membrane. The third piece that we're looking at on this slide here is nNOS. And so we talked about why it's important before, but the reason for it being important at the membrane is really key because the location of how far it can act is critical. If a nitric oxide is signaling from the inside of a muscle fiber, it's not going to be able to get to that vasculature. It's not going to be able to prompt the response that it needs, and it can actually cause detrimental effects. So what we saw here is that following treatment, we can see that we had 25 to 53% of nNOS activity, positive fibers across these muscles. And so what we can also appreciate here is these are two different techniques that we use for measuring each one of these different factors. With microdystrophin here, looking at immunofluorescence, and here we're using an enzymatic activity to show that we have active nNOS at the sarcolemma. And so we were very encouraged to see that the results trended together. We can see that the absolute values for our nNOS here were a bit lower than our microdystrophin, but based on the staining that we're seeing and the background that we have in this method, we're very encouraged that this could be something that could even potentially change over time as intensity of some of these lower fibers that we can really sensitively detect on immunofluorescence may begin to be picked up as well from our enzymatic activity assay. And taking all of this together, what we really wanted to see was, can we put a relationship together between the expression that we're seeing and the drug that we're delivering to be able to actually confirm that what's getting to muscle is doing what it's actually showing here. And so this last piece of data that I'll go through before I hand it off to my colleague Matt is looking at the vector genome copies per nucleus in these muscles. And so in the same muscles where we evaluate microdystrophin expression, we also quantify how many copies of what we're delivering to the muscle with SGT-003 are actually getting there. And so we know that we need to have at least one full copy to be able to have the expression of our microdystrophin. However, the threshold is not really defined on how many do you need to be able to have an optimal effect. What we can see here is that corresponding with the microdystrophin expression, and here an example just being immunofluorescence, we had corresponding levels of our vector genome copies compared to our microdystrophin expression. So we know that as we've seen higher levels of copies, we do have higher levels of expression as well. So we're really encouraged that this could be something that could be saying that across the entire muscle that we're evaluating, we see that we're getting our drug to the muscle, we're getting the transgene in, and multiple copies are present, potentially to be able to even normalize around some muscles that might have lower amounts and some muscle fibers that might have higher. So overall, we can get a well-balanced amount of microdystrophin that could be potentially protective. And so on the topic of protection, I'll hand it over to Matt to go through some of our more clinical data.
Thank you, Pat. I'm going to start us off with talking about just some of the clinical biomarkers that we use to make sure that we're level setting, because I know there'll be a broad group of experience for people who are watching this. As we think about biomarkers, these are different chemicals or enzymes. I think most of us have heard of in the field AST and ALT. These are actually enzymes that are leaked from skeletal muscle if there's injury. You will also hear about them in liver as well, because they can be found in liver cells and leak out from there as well. There's also chemicals such as CK, which in Duchenne is oftentimes discussed. This is, again, an enzyme that is leaked out. Similar to that is titin fragments. To discuss a little bit less, but is also something that is found inside the muscle and is released into the serum and urine when the muscle is damaged. LDH is similarly coming out of cells when there's tissue damage. Now, outside of the muscle, but in the heart, which is also a muscle, is the cardiac troponin I. And this is also an enzyme that is inside these cardiac cells, but gets released when there is myocardial cell injury or injury to the heart cells. Now, those are all biomarkers that we'd find in the serum, for example, we measure as they're released out. In addition to this, there's another biomarker to discuss, which is eMHC, and I'll talk more about it in a future slide. But this is actually expressed in newly generated dystrophic muscle, so we see this more in a biopsy. Next slide. Now, eMHC is elevated in dystrophic muscles primarily due to the muscle breakdown and regeneration. So during a regenerative process, so the point where the muscle would be sort of reforming or trying to heal, the newly formed muscle fibers express this eMHC. High eMHC protein levels in muscle biopsies are indicative then of an active muscle recovery or regeneration. We know in Duchenne's, muscle generation can't keep up with muscle breakdown, and so there's consistent expression of this eMHC going on in these muscle fibers. So sort of as a summary, the upregulated eMHC in dystrophic muscles makes it a potential biomarker for disease and a potential indicator of response to therapies. Next slide. If we stay within our biomarkers, then we can see that in our first 3 patients treated with SGT-003 at day 90, they had a trending that was very encouraging. Serum CK, LDH, AST, ALT, and titin were all decreasing. In addition, that histologic eMHC I just mentioned also was decreased on average, as well as the serum troponin. So the average values had a trend that was encouraging and demonstrated improvement. Next slide. Now, some of these improvements, again, we can see otherwise. Improvements in biomarkers, you can see on the leftmost slide, which demonstrates the microdystrophin that was discussed before, as well as the nNOS. As we move over to the serum titin, which I mentioned before is coming from inside the myocyte or the muscle cell and is leaked out with muscle damage, in our first 3 patients, the baseline was actually demonstrated to be higher and decreased at day 90. In addition to that, as you can see what I just discussed about eMHC, which is the green dots in the slide at baseline, at day 90, we're not seeing a significant that amount. There's a decrease in average of 59% between baseline to day 90. Next slide. Now, those were all very reassuring from the skeletal muscles component, but we do know that a loss of dystrophin leads to progressive degeneration of the cardiac muscle. Cardiomyopathy is, unfortunately, the leading cause of death in Duchenne's muscular dystrophy. And so early troponin levels have been used as a prediction for severe cardiac disease and neuromuscular diseases overall. And early detections of changes in heart-using troponin could inform interventions about disease progression. Now, that's important as we discussed because AAV SLB101 showed a greater cardiomyocyte transduction compared to AAV9 in vitro. That's the graph on the left of a little bit over 20 times in comparison. In our participants, in the first 2 participants who reached day 180, there was a mean baseline increase in cardiac function by EF of 8% from baseline. Our participant 3 had not yet reached day 180, but did also show an increase in EF, ejection fraction. Well, early on, and early in both the disease and number of participants, this was also encouraging data that we're able to see, given that left ventricular ejection fraction has been shown to correlate with clinical outcomes. Next slide. On the same note, in the one participant, participant number 3, who had an elevated troponin concentration at baseline, there was a reduction at day 90 of about 36% as well. And as I mentioned before, cardiac troponin I is released during heart cell injury. There has been evidence that reducing troponin concentrations by over 25% have been shown to have a reduction in cardiac risk of events as well. So again, more encouraging early data that we have. Next slide. And beyond just the biomarker data, the overall safety summary was encouraging. We used prophylactic glucocorticoids alone. There was no need for intensive immunomodulation. All treatment-related adverse events resolved with no long-term effects. They began within days following treatment and resolved within weeks. In addition, there were no serious adverse events and no kidney injury was observed. It's typically called TMA or HUS. For adverse events of special interest, these are events that are called out specifically to be monitored. There was an episode of mild transient elevated of troponin, but the EKG and echo changes were not seen, and there was no clinical evidence of heart inflammation, what people might call myocarditis. I'm going to call it right here that CTCAE grading. This is a grading system that is standardized, that used across studies to be able to identify clinical components. 1 is considered the most mild and 5 is more severe. So for those who are interested, you can actually Google the criteria if you're wanting to have a longer read. Also of note, going back to the AEs, the most common adverse events that were seen was nausea and vomiting, and those have all resolved. There were low platelet levels, or also called thrombocytopenia, observed. 1 had a level that was reaching that CTCA grade 3 level, but all returned to normal laboratory ranges with no clinical symptoms or no bleeding. There was no evidence of blood breakdown, red blood cell breakdown, called hemolysis, and no need for eculizumab, increased steroids or other agents used, and no hospitalizations or interventions around this. There was also 1 infusion-related hypersensitivity reaction that was a little bit stronger. There were a few that happened around the other time. They all resolved. The stronger symptoms were secondary just to a prolonged fever. Again, that was graded as a grade 3, however, but also resolved. In regards to liver injury, there were consistent declines in AST and ALT, as mentioned in previous slides, throughout the course, and no GGT increases. GGT is a liver-specific enzyme that we can measure in the blood as well. So overall, the adverse events were expected and consistent with our AAV gene therapy. Next slide. Now, I know I've spoken very quickly through this, so to sort of summarize, our initial muscle biopsy results for the first 3 participants reaching day 90, we demonstrated an average microdystrophin expression of 110% in Western blot and 108% in mass spec. There was an average microdystrophin percent fibers involved of 70%. Again, that's the percentage of how many fibers were demonstrating microdystrophin, and the average beta-sarcoglycan percent fibers were 70%. The average nNOS fibers, as we discussed before, 42%, and the genome copies per nucleus were 18.7 as well. So all very encouraging results that we were seeing from these initial patients. Biomarkers of muscle integrity also trended in the right direction. The results for the first 2 participants at day 90 were consistent improvements. There were no serious adverse events in the first 6 participants treated with a data cutoff as of February 11th of this year. The most common events were nausea, vomiting, and thrombocytopenia. And there's currently clearance of both UK and Italy clinical trial applications. So we're very excited with this data, and upcoming, we're looking to activate additional trial sites by the end of 2025 and to continue participant screening and dosing as we move forward. And then one last slide before we take questions. So again, I want to thank you all for having us here to talk today. This is a list of our currently active sites and the QR code that should take you to the clinicaltrials.gov posting, which will be kept updated as we go forward. With that, I'll turn it back to Eric.
Thanks, Matt, and thanks, everyone. Great talk. We did have a lot of questions come in, both from the submission before and then as the presentation went on, and I know we don't have a ton of time, so I'm going to try to get to the ones that I think were most common, and then if we have time, we'll kind of drill down into the rest of them. Maybe we can head back to slide 14, because that might be a good place to start. I think slide 14 and 15 had a number of questions around those. I think here, just one thing as well to emphasize, because as patients and families probably saw kind of the increased trial time, but this is an open label study, so expectation here is all patients would receive SGT-003, not placebo-controlled. We did have a number of questions kind of around immune modulation, of whether or not you considered or explored any other. I know you kind of highlighted that as of now, prednisone is kind of the main immune modulation. So just If you could expand on that, how you came to that kind of conclusion for the study design.
Matt, do you want to take that one?
Sure. So looking at, again, preclinical data as well as sort of coming into the space, the feeling was that prednisone, especially for these boys, an increased prednisone amount dosing would be sufficient to look at the safety profile. So that was some of the discussion. There was a more lengthy discussion, obviously, about all the safety components. We've been very reassured with the initial safety data that we have, with the signaling that we're seeing, that this is the regime that we are currently on is correct at this time. But of course, this is early in the trial, and we'll continue to monitor for the duration.
Great. And then we've also had some questions around potential ages of obviously cohort 1 and cohort 2, kind of 4 to 11. Through that of, what are Solid's plans around considerations for older non-ambulatory boys?
Yeah, I'll take that. It's a great question. I think, as always, and especially from the inception of Solid, we've been committed to the Duchenne community and really want to put together a program that's able to get to as many patients as possible. So where the study currently is 4 to 11 years old, we are in the process of evaluating ways to expand that as well. We want to be mindful, especially of safety. And so that's why we're conservatively progressing through different age ranges. But we will be updating the community with more as we have plans to be able to move into different age ranges as well.
Great. Yeah. On the next slide, there were some questions around kind of the variant or mutation restriction, and I think you covered that nicely. But just to, I think, reiterate for folks, some of this being of safety concern at this stage. And for families who are kind of wondering around this 1 to 11, there's a webinar that PPMD previously hosted with the NIH kind of covering this, where a number of the companies came together to kind of explore what they were seeing across different gene replacement studies, looking at this potential immune response to the transgene. The question with that, though, was, okay, does Solid have plans to kind of eventually look at inclusion of these individuals who maybe currently are being excluded from these trials?
I can take this one. It is something that we're currently exploring, and we want to be broad as we think about it, but we also want to progress safely and very sort of stepwise to make sure that we're maintaining safety first. So yes, it is something we're evaluating.
Great, thanks, Matt. And then on the antibodies, we had a few questions here. So first, can you just clarify, because I think between different clinical programs and then there's also commercial products of sometimes the negative for antibodies, there can be a range or a cutoff, or it is a binary yes, no. So can you describe what is that for Solid? And then with that, I think a really common question that we get, of course, is what are you, as a company working on or towards for looking at kind of these individuals who we know have some of these pre-existing antibodies?
Yeah, absolutely. I think one of the key pieces there is that every program will typically have a unique assay and a unique cutoff that they are using for antibodies. So that is an important consideration. With the test that we're using, we don't yet have data to say that we can treat patients who have some level of antibodies that they would still get the benefit that we're hoping for. So with the criteria that we've established, it's really based on understanding from the assay sensitivity, can we detect antibodies or not? So we'll get a titer that comes from the assay, and we'll be able to relay that as well. But it's more of an analytical threshold, just based on normal positive versus negative, than some data that say that we can actually expand out further than there. That being said, we're very interested in looking at specifically that, looking at natural seropositivity, looking at redosing. And so these are actually active research topics that we are working on right now with our R&D team. Specifically, we have an immunomodulatory team, an immunology team who are working towards these. We're hopeful that as we generate more data, especially from the non-clinical side, and learn what the effect can be of the drug with some of these levels, then we can start to reevaluate exactly what the criteria are. But again, like most of these things, we just want to make sure we're doing it in the right day and we're in the right way and that we're data driven.
Thanks, Pat. And I think that redosing question as well, that's one that came up a lot in the questions was, is there an opportunity for redosing? You know, one of the exclusion criteria listed here is that if you've received a gene therapy, whether investigational or commercial, that's an exclusion. So families had questions both around, you know, is there a plan, or does Solid is exploring redosing? And then I think along with that, usually the other question that comes hand in hand with redosing is, do you know kind of durability, and you know, what kind of data do you have around the durability of this transgene delivered by SGT-003?
Yeah, so I think on the redosing side, it's something that similar to natural seropositivity, it's something that we're actively evaluating. We've had interesting data to show different profiles of different antibodies based on your initial treatment that you've seen. Whether you get one serotype, you may have different antibody responses against another. However, they're very early days in some of these research efforts, so we'll look to put together more studies that give us a better idea of what may be possible with different antibody thresholds, especially. But that question of durability, I think, has been the most important one, especially as we evaluate different age levels, age ranges, and younger patients, especially. We want to make sure that this will be a long-lasting effect. So far, this is a very new study for us. These are the results that we have to date just from these short-term time points. I think we'll look to others in the field as well, as they have a great experience to be able to look to see how durable a response can be. And then we'll try to generate our data too to support that.
We also had a question around kind of I would bucket into this prior treatments around cardiac meds, knowing that you do have a cohort in that 7 to 12, and that many of the boys with Duchenne may be started on a cardiac med like an ACE, kind of around age 10. Is there any exclusion around that for cardiac meds?
Oh, no, there currently is not, for standard of care type cardiac medications, any exclusions around that.
And then I guess one point within this that I wanted to highlight, just so families are maybe aware or noting, is, you know, you have the washout for a number of the exon scare therapies, givinostat, and I think specifically here to point out vamorolone, because you require a stable steroid regimen, but that is specific to prednisone or deflazacort. And so if someone was on vamorolone, this would be part of that 12-week period of needed to have washout and transition to something like a deflazacort or a prednisone, correct?
That's exactly right. And just to kind of emphasize the reason for it is just understanding kind of what the impacts of each of these different therapies are. I think vamorolone is very interesting and new. As we look to things like natural history comparisons, this is where we have more data. So we're looking to be able to learn as much as we can from the study, and then use factors like this with prednisone or deflazacort to really compare against as we're in the early days.
And then can we maybe jump down to slide 17? And I don't know if you'll have a hard stop at 2. I might have a few questions to go a little bit over if possible, just as we unpack some of these.
Of course.
So first of all, I think it's really helpful to have these multiple methods for showing expression, just so families can help understand kind of what we're looking at. And I think maybe another way too, as we talk about these to help orient families, is, you know, these top two very much we're kind of looking at, you know, how much of the microdystrophin is present. The bottom piece is more of the where and the biodistribution. So these are both really important components in kind of understanding when we look at any of the dystrophin restoration therapies that are kind of in the space, for our ability to kind of interpret them. And Pat, I think you did a good job. And I think as a reminder to folks, again, you touched on, there is a normalization that you're doing, because I know I had questions from some of the families of how do we get to something like 110%. So I appreciate you kind of breaking down how that works. One of the things you said for the mass spec was you can look at something that is unique to the microdystrophin that maybe isn't present in dystrophin. Could you explain that a little bit? Because I could understand if families might be a little bit confused, thinking, okay, well, what would be in a microdystrophin that's not in a full-length dystrophin?
Absolutely, and appreciate the opportunity to clarify that. So In a full-length dystrophin, we know exactly what the sequence is, and that's going to be what you have naturally occurring. If you don't have Duchenne, you're going to be expressing that full-length protein or Becker, a full-length sort of protein. When we look at a microdystrophin, we take the full-length sequence, we take the full-length protein, but we take pieces out of it, which means that some of the combinations of pieces within a full-length protein are going to be coming together for the first time. And so because we have that gap, now we can look at what is this novel junction. So we call them novel junctions because they're coming together in this microdystrophin specifically, and that's what we can measure. So we know it's not naturally occurring. It's only in this microdystrophin.
Great, thank you, that's helpful. And then on the next slide, I think this is just helpful because there was a lot of data presented, and so I hope and encourage families, take some time when the recording comes back to listen to that or go through to try and make sure that you're seeing everything. But essentially wanted to highlight here that as families look at this type of data and as they look at some of the biomarker data, these are additional things we look at to help try and understand and build evidence to predict of kind of what we think is likely with these therapies. So here, you talked a little bit of the role, and I think it's important to understand for families that, the previous slide showed how much microdystrophin is there and where is it at. And this helps to show, functionally, what is the microdystrophin doing? Dystrophin has all these roles that it plays in stabilizing the muscle membrane, and some of it is interacting with some of these other proteins. And so this is showing some evidence that the microdystrophin is not just present, but maybe doing some of those functional roles. Could you maybe talk a little bit, though, why I guess we're going to see maybe different numbers? So you saw dystrophin expression of 110% bio-Western, and then you have the different numbers with the percent positive fibers. And then as we look at some of these other things like the nNOS activity or the beta-sarcoglycan, they're not always, exactly the same. There's variation there.
Yeah. And so when we're looking at that absolute protein value, we can go above 100% because that's how we're measuring an absolute value. When we're looking at immunofluorescence of percent positive fibers, the maximum you can get here is 100%, and that would be getting to the entire muscle. So that's where we know that those absolute values and the percent positive fibers can be different. As we look at each of these different markers, I think we'll have different sensitivities to changes that we're looking at, because things like beta-sarcoglycan, while it is expressing in a dystrophic muscle, it's at a low level. So we're looking to see now how much is being restored. Similarly to an nNOS, this is something now that's mislocalized from the membrane. It's really intracellularly now rather than at the membrane. So again, we're looking to see it restored. I think this is why we see some of these differences in the absolute numbers in these different methods. But we hope to also see how they'll change over time as expression may also change as we look to things like that 1-year biopsy time point.
And then one of the questions we had kind of following this was, is there any functional data? And so I think, at this point, too early for the functional data, but maybe you can talk about your plans. And you had some of what you're going to be capturing in the earlier slide, but maybe timelines to when the community might be able to expect hearing from Solid on that. Because again, this is gives us some evidence and points us in a direction that we would hope translates to functional benefit, but we need to collect that data, obviously.
Matt, you want to take that?
Yeah, so as we just sort of mentioned before, and Pat, if you don't mind going up to the trial design slides, just as a demonstration, we are collecting functional data in the trial. Those data points are looking at day 540, so a little bit further down the ways in this initial day 90 microdystrophin expression data. In regards to when the community will know, so we want to be transparent and engaging with the community quite a bit. There are a lot of factors that determine when and how we are able to discuss that data going forward, but certainly we will continue to engage overall with this and as we can otherwise. I think Pat called out early that the idea that some of our data, especially the microdystrophin data that we presented today, we analyze in a batch process. So as we come forward with data, it will be in groups rather than patient by patient. I just want to call that out for the community so I don't want to set expectations that there are going to be every patient that we're going to hear more about. But as we're able to talk more, we certainly will be doing that.
Great. And then maybe as we hop down to the slide 25, which had some of the the cardiac data. While we do that, the question did come in just on the number of biopsies that are done during the course of this study for each individual patient.
3 total biopsies, 1 pre-treatment, 1 at day 90, and then 1 at 1 year.
Thank you. And then I think just because, again, a lot of data here, which is great. And I, again, encourage families to go back, listen. Again, just wanted to highlight, looking here at the data, early data on the cardiac, maybe just a comment on the age of these patients, because I think that is important as we look at cardiac function with, again, 2 5-year-olds and a 7-year-old versus as you continue to look in the future towards, older patients where this, we may expect kind of differences in terms of ejection fraction.
So thanks, Eric. And that's right. These are relatively young, very brave boys that participated in this trial. As we allude, we're, looking at older boys to partner with us as well, where we feel that, we're at a higher likelihood, frankly, of seeing impaired cardiac function, impaired versus normal. I think, we are dedicated to investigating the safety of SGT-003. And for that reason, we looked at very carefully what's going on with the heart, especially because we know that the hearts of these boys can be, can show that injury even this early. And so that's really the reason why, we developed a database, or starting to develop a database of what the troponin looks like, that enzyme that's released when cardiac muscle cells are damaged, or looking carefully using echocardiography, using sound waves to create a picture of the structure and the function of the heart. And so I think we share that we were very encouraged by what we see has been some encouraging signs of safety. That we didn't see any myocarditis or anything like that, or evidence, and that when we've looked at the structure of the heart, we actually saw, as opposed to concerns, we actually saw potential signs of optimism that this could actually might be something that could be of benefit for these boys, something that we need to investigate, and we feel compelled to investigate more.
Thanks, Gabe. And then maybe we can go to the last slide. And I do want to say, I mean, that is something I know Pat Furlong has long shared with our industry partners, that, that is something we want to see and be monitoring with all the investigational products, is to understand what's happening with the heart, even at the young ambulatory ages, because it's going to be important as the boys get older. So maybe one more, the one that had just the information on the sites and the contact, just as we kind of wrap up here. So I do appreciate you all staying on a few minutes later as we got to some additional questions. I know that there were some more coming in, so again, please send those to us at PPMD, and we'll pass those along to the team at Solid. But just wanted to thank you all for coming on and presenting and sharing that data and walking us through it. And again, for the families, there's good information here if you're looking to learn more about the sites that are participating and how you might be able to participate. We will have this up on the website, hopefully within the next week. And again, send any questions you have into us as we go. So just thank you all for joining us, and thanks everyone online for joining us for the webinar today.
Eric, thank you so much for creating a platform for sharing innovation.
Thanks, everyone.
Go on.